In this lab station, we're going to take a look at paper chromatography. So I'm going to follow the example. We're going to put a pencil line at the bottom. I'm going to use a ruler to make that a little more straight. And then we put a pencil line going up the middle. And the chemical or mixture that we would like to separate, I'm going to use a black marker here. I'm just going to put a little dot, tap it multiple times on the intersection of those two lines. Now what I need to do is clip that here and hang that in my cup or beaker. And now what we want to do is make sure that the water level is below the pencil line where my dye is located, but it has to be touching the paper. So I'm just going to squirt some water in here, nice and slow. So now that water is traveling up the paper, and when it hits the dye, it's going to carry the dye with it. So if the dye and the water have very similar polarities, the dye is going to stick with the water and travel up the paper. And eventually what we'll do is we'll measure the distance from the pencil line up to the water and the pencil line up to the distance where the dye traveled. So while we are waiting, I'm going to take a look at this red dye here. So I'm going to measure from pencil line up to where the red dye is located. So to me that looks like that's about 3 centimeters. And if I go from pencil line to where the water was, that's about 5 centimeters. So if I divide those two numbers, 3 divided by 5 would be 0.6. So that would have an RF value of 0.6. Alright, so if we take our black marker sample out of the water, I'm going to go ahead and calculate the RF value. You can see here that this black marker is actually a mixture of several different colors. We've got a gray color down here at the bottom, and then a little bit of a light yellowish green color here in the middle, and then a blue color up top. So I'm going to go ahead and calculate the RF value for the blue color. So it looks like the blue traveled 4 centimeters, and it looks like the water also traveled 4 centimeters. So in that case, you've got 4 divided by 4, which is 1, which basically means the dye was really attracted to the water and traveled the same distance as the water. Whereas the yellow looks like it's closer to about 3.5. So that would be 3.5 divided by 4, whereas the gray has an average distance of 1, so the gray would be 1 divided by 4, which would be only 0.25. So that means the blue has polarity and intermolecular forces very similar to water, whereas the gray is probably less polar, less attracted to the water, so it stays closer to the bottom. We can see from this sheet here that paper chromatography can be used to separate the colors found and the different pigments found in different leaves. So on the right here we have a red oak leaf, So it's a little bit faded now, but this leaf used to be green. So we've got some green color up here, but then they turn brown in the fall because there's another pigment called tannin found in the red oak leaf, whereas a maple leaf you can see has lots of different colors here. Over time they all just kind of turn brown, but the major pigment would be green, the chlorophyll, but here we had some blues and yellows and oranges, and that's why maple leaves are so pretty in the fall.